It's coming up on the 50th anniversary of the discovery of the heat shock response by Ferruccio Ritossa. It'll be in 2012, and those of us who benefited directly from that discovery in various ways have been thinking about what Ferruccio Ritossa means to us in our careers and our research. And so for me, I didn't know about the heat shock response or Ferruccio Ritossa for several years in my research. So I was an assistant professor at the University of Connecticut, and I was studying viral replication, and I stumbled across this observation that analog substituted proteins, amino acid analog substituted proteins, could induce a new set of proteins, a small set, 6 or 7, but it was a dramatic change in the pattern of proteins synthesized by cells. And it turned out it had nothing to do with virus infection. It had everything to do with the cell accumulating amino acid analog substituted proteins. Well, I didn't know what this meant. I didn't know whether this was some strange aberration of the cells I was using. I had no physiological context to put it in. I didn't know what the function of these proteins might be. And then a friend of mine who worked with Drosophila stopped by, and we had dinner one evening, and I told him what I was studying, and he said, well, I think you may be studying the Drosophila heat shock response. And this was Thomas Klein, who was a fellow graduate student at Harvard during my grad student days. And Tom was the one who really put me on to the possibility that these might be Drosophila heat shock proteins. It was really the first I'd heard of them. So I started reading papers, and I found Ritossa's paper, the 1962 experience paper, showing that really environmental stress, signals coming from the environment, could dramatically change gene expression in animal cells. So that really made me think that my analog substituted proteins were somehow related to this heat shock response. And then in 1982, we had the first international meeting at Cold Spring Harbor, where all the scientists studying the heat shock response in different organisms got together and traded information and traded their stories on, how they got into the field and how they found these proteins, and everybody talked about Ritossa's discovery. And in fact, Ferruccio Ritossa was at that meeting, and he was applauded, and I think he was given a Cold Spring Harbor T-shirt, and and people thanked him for for this remarkable discovery. And for me, it was both the discovery and the community that came together to study these proteins that made a huge difference in my career, because suddenly I had other people to talk to about the response and the proteins and what they were seeing and what they were thinking about. And it really became a community effort to try to understand the regulation of these heat shock genes and what the proteins did, how did they function, and ultimately how did they work in the physiology of plants and animals. So for me, it was the discovery and also a chance to bring my observations on analog substituted proteins and the hypothesis that animal cells could sense the presence of damaged proteins and respond to that with new gene expression, to put that in a broader context, and to have people, colleagues in a field to talk to and work with. So that's what I'd like to say.